That started. Progress, you've all been warned. All right. Well, hey, everybody. Thanks for showing up on such a stormy night. It's great to see you all here. We're here to talk about the state of the Saak here in the northern Taiya Inlet. I'm Ruben Cash. I'm the environmental coordinator here for Skagway Traditional Council. With us, we also have our program assistant, Colton Belisle. He's going to be getting up and talking a little bit. And we also have our project lead for the eulachon population monitoring project, Meredith Pokart. She's come all the way up here from Haines to come and give us some great results and some data. So if anybody has been to any of my talks, you know that I love a route map. I just absolutely do. And first you have to get your equipment to work correctly. So let's see if we can make this work. Maybe. There we go. Okay. So here's our agenda. We're going to start out, we're going to give a little bit of a background, right? We want to look at that 10,000 foot view of what the eulachon is, what its biology is, where it is, all that fun stuff. And then we're going to talk about the cultural significance of this fish, because it really has affected a number of different groups all across its range. And it's got a pretty large range. So that's definitely something that we want to talk about. Then we're going to jump in. We're going to talk about the status throughout its range, including some of the status we have here in Alaska. We'll go over the previous efforts that have been done specific to this project started by the Chilkoot Indian Association. And then we're going to talk about environmental DNA. It's just fantastic methodology. It's a really cool way to look at a critter and its population. Then we'll go over and we'll look at some photos and some reports from the 2023 field season. And we'll look at some results and a little bit of a regional picture. All right, everybody still on board? All right, let's jump right in. What is the eulachon? Well, the eulachon goes by a lot of different names, right? I call it eulachon. I also call it Saak, which is the traditional Tlingit word for it, but it goes by a lot of different names, right? Eulachon is probably a little bit of a mispronunciation of the original Chinook jargon, which was eulachon, right? And the Chinook jargon was spoken by, it was a pigden, so that different indigenous peoples and settlers could speak with one another. And so it was referred to in that Chinook jargon, that pigden, as eulachon. And you can see there are a number of different ways that this was mispronounced, including the all popular hooligan, which also comes from that eulachon. We also call it the Saak, right? That's the Tlingit word for it. The savior fish, the candlefish, the fathom fish, the smelt, and this is just a little bit of the names. If you look into the etymology of this particular fish, it has a really rich history of being named. And the reason for this is because it touched a lot of different cultures all throughout its range. It was a very, very important fish. They call it the savior fish, right? And they call it this for a reason. And we'll get into some of the reasons on that. But I like the Greek. That's my favorite, right? I love me some scientific news because it really tells a story about what the fish is. Thales pacificus. This translates directly to rich fish of the Pacific, right? And it's not what you're thinking. It actually has more to do with the fact that it has an awful lot of a very high oil content. And that's why this was called the savior fish, because these fish came up early times of the year and they provided this fat source, this protein source for the first time all winter. Everybody's been starving, the people, the wildlife, everybody's been kind of going through this little bit of a short famine of the winter time when there's less food available. So the savior fish comes through and provides this sustenance to all the people, right? So let's look at the range. And I've created this really cool map. We're going to start really zoomed in because we know these two rivers have eulachon in them, right? We all know where we're at here. We all recognize our spot. So this is the Taiya River and the Skagway River. So this is the data set that was pulled from a national hydrography data set. It's all the streams, all the canals, all the rivers, and all of North America. It's a massive data set. If you load just one section of this into your computer goes and freaks out and doesn't really work for a little while. So you have to kind of pull data from it. So that's what I did. I did what's called an SQL search. Basically searched through and added some keywords. And I was basing this all off of a paper written by a gentleman named Wilson back in 2006. This was a synthesis paper that talks about everything we knew about eulachon up to that point, including the different rivers where we knew that it was. And so he was stating that there were 30 to 139 North American rivers where we know that eulachon have been spotted. And so I took all those rivers and I typed them in one by one, and came up with this data set where it just pulled the rivers where we know we have eulachon, right? So there's a caveat to this thing. As we zoom out into the greater regional map, you have to remember that even though the whole river is being shown, this is not where the eulachon are going. The eulachon are going to be using the tidally influenced reaches of these rivers. So in Skagway River, that's up to there. But if I'm zooming way out and showing all of North America, you're not going to be able to see that data point, right? So I included all the rivers so that you can see them. But just keep that in mind as we zoom out. We're not actually seeing eulachon all the way up in British Columbia on the Columbia River. They just don't go that far. So let's zoom out just a little bit more, show this closer regional picture. Here's a lot of the rivers that, are we actually sampling all of these rivers that are shown there? Every single one of these rivers is shown. And what I want you to notice here, notice the estuaries on these rivers, right? Notice there's this kind of discoloration that comes off all these rivers. This is all freshwater. This is really important for eulachon in their larval stage because that freshwater is keeping away some of the predators that rely on a higher saline environment, right? Some of these bony fishes that need to have a lot of salinity, a lot of salt in the water, they're going to stay away from these estuarine areas. So this kind of provides this little bit of a nursery for these larvae as they passively drift out of the river. So you can see all the ones that we're showing here, they all have these plumes. You know, the Taiya and the Skagway, definitely in all of these, we're seeing these plumes. So everybody on board? Everybody understand the data set and its limitations? Okay, so let's zoom out and let's look at the whole thing. Takes just a half a second. There we go. All right, so all the way from the Aleutian chain down to the middle of California, we're seeing eulachon. There's 66 rivers in Alaska. There are 30 in British Columbia. We've got 21 in Washington, 16 in Oregon, and 6 in California. And I think a lot of the reason that we're seeing this big difference in the number of rivers has to do with coastline, right? There's a lot more coastline in Alaska than there is even in BC. A lot of BC's coastline is occupied by Southeast Alaska, so it's a smaller coastline. Washington, of course, has a smaller coastline, but it has the Puget Sound, so there's more coastline there, and so on and so forth. So we also know that we have 3 distinct populations, right? They've traced salmon genetics down to the river, and we can definitely say this river has this specific genetic group. It's not the same with eulachon, but we do know that they have these 3 distinct, they're distinctly different from one another from a genetic standpoint. So we've got the northern, which is all of Alaska, the Gulf of Alaska, not including Southeast. We have the central region, which is Southeast Alaska and everywhere in BC except for Fraser River. And then we have the southern group, which is everything south from there. So what do we know about the eulachon? We know that it's a forage fish. You guys all heard of a forage fish, right? You guys all know what forage fish are all about. Forage fish are a really important link to trophic levels. You guys know what trophic levels are? When I talk about trophic levels, trophos means nourishment. So I'm talking about nourishment levels. And I'm sure we've all heard of primary producers and primary consumers and tertiary consumers, all that stuff. Herbivores versus plants versus carnivores, that kind of thing. Well, when we're looking at different trophic levels, the bottom of our energy, where all of our energy comes from, is coming from the plants, right? Those are the primary producers. In an ocean environment, these things are microscopic. We have some macroscopic algae out there, but a lot of what's being eaten, a lot of where that energy comes from and gets transferred is in this microscopic spot, right? So these predators up at the top aren't going to go out and eat some zooplankton. Maybe the baleen whales are probably going to be eating a little more of the zooplankton. But the forage fish are what are going to be consuming this, taking this energy all the way from the bottom and being able to transfer that to something that can eat it. So it's a really, really important link to where most of the energy is. And what we found is that when forage fish have any sort of a collapse or any sort of trouble with their population, we start seeing this cascading up and down trophic levels. Back when we had the blob in the Pacific, we had a whole bunch of shorebird and waterfowl die-offs, right? We had auks and murrelets just showing up in the thousands on the shore. And what we found is that it's because forage fish were falling out from this system. And that energy no longer had a way to transfer up to those birds. So we have these collapses. So it's a really critical part of these trophic levels, a really critical link. The other thing Has to do with monounsaturated fatty acids, or oleic acid. These guys are eating little zooplankton that are full of these little fats, right? They're living in these cold northern Pacific waters. And so all the critters that are living in the water need to have some sort of insulation. So this oleic acid is really, really critical. And so that stuff transfers to these fish. Now let's look at the salmon lance. It's about 3 to 6% of that good oil. If you look at the capelin, it's got about 2 to 10% oil. But look at this guy right here. This is the bacon explosion of the sea, 18 to 20% oil. I mean, that is a lot of oil. That's why we call it candlefish. You can dry that thing out, light it on fire, and it will burn from that 18 to 20% oil. Pretty significant. The only other critter that I know that rivals the eulachon in terms of oil is the Pacific lamprey, also known as the bacon cheeseburger of the sea. Yeah, so pretty critical because of those fatty acids, and because of that, it's that important early season run for a lot of wildlife that have been protein starved, that rely on those monounsaturated fatty acids in order to make their living. So, what eats them? What doesn't eat them? My goodness. This is also pulled from the literature: white sturgeon, spiny dogfish, sablefish, salmon, sharks, arrowtooth flounder, all species of salmon, dolly varden, halibut, cod, harlequin ducks, pigeon guillemots, common murres, mergansers, cormorants, gulls, baleen whales, even orcas, dolphins, harbor seals, sea lions. The list goes on. This is a really critical food source for a lot of critters. And so we have this phenomenon that happens called a grease wave. As the eulachons are coming up all the inlets and the back channels and the fjords, they're being followed by these critters who are seeking out a food source. And so we see this wave of wildlife coming up. And we rely on this when we're doing our eulachon population work, because we can count on the wildlife. We can go out there and we can count, okay, I see 100 gulls today and I see 500 gulls the next day. Something's happening, right? They're following some sort of food source has shown up. And so we're using our wildlife observations to pinpoint the run onset, the run intensity, the duration. Sometimes we can even follow the gulls up to likely spawning sites and different rest stops. So the grease wave, what a cool and significant phenomenon that happens with the eulachon following that oleic acid. We know that they're anadromous, which literally translates to up running, right? And we understand that anadromous means they're living both in the freshwater and in the saltwater. And there's an evolutionary advantage to this. A critter can take advantage of 2 different types of environments for whatever their needs are at the time. It's freshwater systems have relatively few predators, right? So it's a great place to raise your kids because they're going to be in a relatively safe place. But there's not really enough food there. There's definitely not going to be enough of those fatty acids. So they then transfer out to the ocean to put on some weight, and then they bring that back up. They're semiparous, just like salmon are, most of the time. Just like with any science, there's no absolutes. There's always that depends. But for the most part, after they spawn, they're going to die. So they're bringing those ocean nutrients back into these relatively nutrient-poor environments and enriching them and feeding all those wildlife and feeding the people. Unlike salmon, though, eulachon don't have any sort of fidelity to their home stream. They're opportunivores. They're going to find the best habitat, and they're going to follow their friends to go to that good habitat. So they'll switch rivers if conditions just don't look good, right? So reproduction and spawning. In the egg stage, we know that they spawn from January to May. It's usually coinciding with spring river pulses, right? They're looking for that glacier melt to come out and all that fresh water to go into the estuary so that their larvae have a good safe haven to go and hide in. 21 to 40 days of incubation. The females are laying somewhere around 30,000 eggs, and they attach to these little sand particles. They look for sand particles and small substrate, and they use this little sticky goo with a peduncle to attach their eggs onto those grains of sand where they develop, and then they hatch out. Once they hatch out, boy, these things are beautiful, right? Doesn't even really look like a fish. So as you can imagine, this is kind of a helpless little thing. They act like plankton. They're just passive drifters. They've got themselves a little food sack so that they don't have to go out and look for food, but they still have a lot of development to do. So that's where that estuary comes in. It's a really important piece to their ecology. they need to have a safe place to develop. It's not like salmon where they're in the interstitial spaces after they hatch out and they have a safe place, a safe haven. So they're relying on that good intact estuary and all that fresh water to keep the predators away. And they're passively drifting from their spawning site out into the estuary. And from there, we don't really know. We've done a little bit of studying. We've done some larval outdrift studies, but more research definitely needs to be done. I guess they're doing some research down in BC, looking at the distribution of these larvae after the spawning and after they hatch out. Could be good research to do up here too. I think there could be some opportunities to look into that. And then as adults, we call them the fathom fish because they love the deep water. 625 meters, I think they found them, way down there. They spend 3 to 6 years in the ocean, and they're out there, they're eating that krill, right? They're trying to get in that intake and pack on the pounds, pack on the oleic acid, and then they return to freshwater to spawn, and they're, again, going to choose that river with the best conditions. So with that, I'm going to ask our program assistant, Colton, to come up here and talk to you a little bit about the cultural significance of the fish.
All righty. So historically, eulachon have been eulachon have been both an icon and a staple of both Tlingit culture and Tlingit culinary traditions throughout Southeast Alaska. These fish are revered for their high oil count, as we went over, their high fat content, where known for being the candlefish that's used for all these different, they're used for medicine, light, energy, all sorts of different uses that they kind of fill. Their role in the Tlingit diet and their trade value. So eulachon are usually caught in the late winter to early spring when they come up. And they would catch these fish by laying a variety of traps, hooks, hand nets, and dip nets. As you can see here, we have in the top left, there is a halibut hook that would be traditionally used, more of a smaller size for eulachon, but this is what we traditionally used until iron became more commonplace throughout these cultures. And then once iron came in and started being used, they would switch to rakes that they would use to kind of scoop all these fish up out of the water just onto shore. As you can see, they tend to just pile up in the rivers, and these rakes would just rake them up onto shore for easy harvest. They were easy to collect using dip nets as well, traditionally made of sinew. And after the introduced, and then this is kind of this collection of evolution in resources. As resources came out, these fish could only be harvested more and more effectively. One of the primary uses of them, as we've gone over again and again, is their oil. This oil was rendered and was used as a sauce, as a butter, as medicine. It was used as a preserving agent. It had antibacterial properties due to the oxidizing agents in it. And it was used to preserve foods that usually would spoil pretty quickly. It was used to preserve berries, fish eggs, crab apples for use in the winter. So not only could the fish themselves be eaten whenever, they could be preserved themselves for the winter, but they could use to preserve all these other options that they could bring into the winter with them, where they would normally be going hungrier throughout the year. And then when it comes to traditional medicines, they would be taken orally for ailments such as arthritis and tuberculosis. They would be rubbed on skin to treat rashes. And since it's so high in vitamin A and iron, it could be used for colds and all sorts of other minor problems they might be facing throughout the year. The Tlingit manner of gathering this oil typically consisted of digging a pit that they would dig a pit along the shore, and they would fill it with this fish and bury them and let them ripen over the course of 10 to 14 days. Now, after this period, the fish would be dug up, they'd be placed in a canoe, and they would typically be an older canoe, half submerged in the sand, being filled with water, and then this would be used as like a makeshift cauldron. This water would be filled with heated rocks to kind of boil everything together. And over the course of several hours, using a cedar bark scoop, they would be used to just kind of keep pushing this water back to the boat, letting the oil come up to the front. And this would be repeated until they have their oil all harvested from their fish. And then the remaining fish parts that did not get kind of rendered into oil, all leftover fish that they had would be used to stomp. They would stomp this leftover fish and keep reheating it until they had that rendered into kind of a paste or all the more oil that they would manage to get out of it. And this would be continued until they couldn't get any more oil out of whatever fish was left. And then moving on to the fish itself as a vital source of food, they would eat the flesh fresh. They would be skewering the fish and roasting them over spits. And this is the fish itself was usually eaten fresh if it wasn't going to be used for this oil. And then any excess fish would kind of get the preservation treatment. It wasn't a consistent fish. It was kind of eaten raw, whatever was eaten would be rendered and used for year-round uses. Because this seemed less as a food source, more of a commodity because the oil was so valuable, both for conserving other foods, for medical, and as we're about to get into, its trade value. The preservation would be done by cleaning the fish and cutting it into slivers and air drying it for a day or two, and then smoking it for a couple of weeks before being stored in baskets. Male eulachon could be kept for up to 2 years in well kept conditions, while female eulachon were harder to store due to all the eggs in a row that they had in them. And then culturally, the Tlingit and many other tribes would use this food as a major food and gift during potlucks and ceremonies. Eulachon oil would be drank in grand quantities during Tlingit celebrations, and many families would share the oil that they made with extended family members and communities that would gather. When it comes to the fish themselves, they carry their own ceremonies in many cultures, and various cultures had different rituals for the first fish that they would catch each season. With the chiefs of important clans such as the Waikato holding the honors of plunging the first net into the river, catching the first fish, where they would greet the first fish caught with greetings such as, "Welcome, friends. You have come to bring wealth to me and my tribe. Welcome, oil women. Therefore you come that we may eat." And these Shimshan would prepare the first cut eulachon in special manners using special recipes, accompanied with its own ceremonial outfits, including men's traveling caps and mittens. And after cooking, it was important to some of these tribes to partially burn the fish before they would start their feast, followed with a prayer. And then economically, the oil was a vital trade item among tribes that did not have access to eulachon fishing sites. It was so highly prized by many cultures that the Northwest coast cultures would trade it long ways into the interior along grease trails. Locally, before the gold rush, generations of Alaska natives used the Chilkoot Trail as one such trail to get into the interior. The Tlingit, Haida, Nuka, who called Vancouver Island home, did not have eulachon rivers of their own. Either they didn't have literal rivers with fish in their area, or they did, but they, as a tribe, did not have access to those rivers. They were controlled by other tribes and chiefs. So it was important that these tribes were able to trade for these resources, both the oil and the fish. Haida and Tlingit fishermen would meet the Shimshan tribe at the mouth of the Nass River, where the Shimshan would fish to make their trades for the oil. The Bella Kula valued both the fish and the oil as trade items, second only to salmon, traded among the Bella Bella and the Chilcotin. And that's all I have. And I'll pass it back to Ruben to go over the status of fisheries and the eulachon.
I just want to go back to this slide here and about Skagway these fish, because we otherwise wouldn't have known that there was a trail up and over that pass to get to the gold fields. And perhaps they would have found the deep harbor that was here. But I think the eulachon really led the way for Skagway to come here. So I just wanted to throw that out there. At any rate, let's talk about the status, right? There have been a couple of commercial fisheries. I can't remember the names of the fisheries in Alaska. Is it the Nass River and the Stikine? Do they have fisheries on them? The Eunuch. The Eunuch and the Cocoon. Okay. Yeah. But south of here, the only fisheries that I'm aware of were the Fraser River fishery, right? Fraser River out of Vancouver, BC, and the Columbia River fishery. Again, they didn't go all the way up and but we have these long old data sets that show us numbers over time. The Columbia River fishery goes back to 1938, and the Fraser goes back to 55. And you can see that there's this trend, right? And both of them, numbers go up and down, but then all of a sudden in the mid 90s, everything just collapsed and they closed both of these fisheries. 94, I think, for Columbia, and just after 95 for the Fraser. And notice that on the upright here, we're showing the eulachon catch in tons. And so the earliest records that we have show that it was about 200 tons of eulachon. But they've done some research and they found that indigenous catches were many times that, but in the thousands of tons were caught prior to colonization, prior to contact. So that should tell us something. So the distinct southern group, right, all the ones in red, they're now listed in the USA as endangered under the Endangered Species Act. And in BC, they're listed as endangered along the south coast. And then on the central coast, they are a species of special concern, right? So what are the factors? What are some of the things that are going to cause these declines? Well, obviously, for an anadromous fish, this is going to be a problem, right? You're not going to be able to get to your spawning grounds. If you're on a low gradient river, you've got a dam that's down there and that tidal influence. Yeah, we've got dams and we've got fish ladders for salmon, right? But salmon are a charismatic fish. There's a lot of money associated with that. They're not thinking about the less charismatic fish like the eulachon or the Pacific lamprey. And indeed, even in places where we have these fish ladders, they find that both of those fish aren't able to pass those dams. And so we're cutting off a huge amount of their spawning grounds by blocking their anadromous, right? What's another thing we love to do? We love our navigable waters, right? We like taking our boats up in our rivers, and to do that, we got to dredge things out. And what are eulachon spawning in? They're spawning in that fine sandy substrate. So when we take that out, they no longer have a place to spawn. We're just digging out the center of the channel, and we've got some shelves on the side, right? And the eulachon run comes up, and they're going to spawn there. What happens when there's an underwater landslide because the banks run steep? We no longer have a place to spawn modified navigable water channels. What about the estuary itself? Anybody ever been to the Duwamish waterway in Seattle? This is just south of Elliott Bay. This is a river. This is the mountain of a river. It's not even recognizable as a river. In fact, there's only one piece of actual shoreline left in the Duwamish waterway. It's just about a quarter mile up, and it still doesn't look that great. So even though we're pumping fresh water out, we've got no place for them to spawn. Another thing that's really significant is the sea surface temperature anomalies. Anybody remember the blob from back in 2016, I think it was, where we had this amazingly high temperatures, I think 60 degrees Celsius above what is normal. And this causes some problems. Remember, the eulachon are going out there and they're eating these monounsaturated fatty acids. They're eating these krill that normally live in this cold Pacific Ocean. Well, when the blob was happening, we started finding these tropical species of phytoplankton migrating up into these waters. Are eating these things, and they're not getting the ecosystem out of balance. Ocean conditions are definitely going to affect that. They've got a lot of things that are kind of going against them. What about our status in Alaska? Well, wait a minute. We know that there's a demonstrated aversion to excess noise, and that I think causes a bit of a problem because we really don't have any protection for them up here. They're not listed anywhere except, is it the Cook Inlet? Do they have any listings in Alaska at all? There's no protection in the state of Alaska at all for these fish, even though we know that they're in decline in a major portion of their range down south. We know that these things have been happening, all this development, hydroelectric dams, changes to the estuary, changes to the river environment, poor ocean conditions, but we've got nothing to protect them up here. There is this demonstrated aversion to excess noise. We've heard indigenous stories, all this traditional ecological knowledge that talks about singing a quiet song when the eulachon come up the river, and to not disturb the first scouts that come up in there so that they don't decide to go to another place. We've got tens of thousands of years of these stories being passed down telling us that this fish has an aversion to noise. And we do have a little bit of information from the study that we've been doing, like the Chilkoot River in 2015 or 2016, there was some pile driving happening at the ferry dock, and they abandoned the Chilkoot. They just didn't enter the river that year. And then in 2019, the lower reaches of the Chilkat, they were doing some blasting along the highway, and all the fish decided to come up here to the Taiya River. And that's great and everything. But if you think about that map that shows the Chilkat and that big, beautiful, protected estuary, and then you look at the Taiya and you look at the size comparison to it. So sure, we get fish up here in the Taiya River, but how well are they going to survive in that reduced estuary? It's not as good as the Chilkat. If they had spawned in the Chilkat, we probably would have had a lot more survival. So this can be an issue. They're not commercially important, but the cultural ties between eulachon and indigenous people all across its range really shouldn't be overlooked, right? And so is that the importance, that ecological importance to all these the early run to the wildlife coming through. So it's a little bit of a pickle, you know. If we want to provide any protections for them, we need more data. We need to be able to prove that they have this negative effects happen when we expose them to excess noise, things like that. So, review of efforts. You want to talk about the Chilkoot Indian Association's early efforts? I'm going to pass this on to Meredith.
Yeah, thanks for having me, guys. Okay, so yeah, kind of there's also kind of an overall lack of knowledge and information about eulachon across their range. And so in 2010, the Chilkoot Indian Association, things were starting to get listed as threatened. The southern distinct population that sort of mentioned was listed in 2010. And the Chilkoot Indian Association was like, well, we have no data on Chilkoot. One of the main so in 2010, we created the smart recapture study. So kind of it's the first stab at getting some sort of population data, engaging the overall size of the run, and then kind of more western data. So basically how this works is we so at the mouth of the river, there's a bridge there. We put a trap in the river. Not actually a photo of the trap, but then collect fish there, add a close thing, give them a flip, and then upstream there's a weird Chilkoot River, pretty distinct marker in the river. So all of this is our recapture reach. It's also kind of there's also a culture campus right there. Where all the spawning happens, like not coincidentally, like it's like a very popular eulachon location. This is where we recapture. Through that ratio of the clip fish and the fish and again. So this was a good method. We started doing it 2010. Let's actually update this. We're still doing it even in 2023. But it's really only works on the Chilkoot River. Now it's kind of like a lot of different eulachon spawning rivers, and there's really not another one where I would actually do a market capture study. Pretty unique. Chilkoot wadeable, pretty short, landmarks. So it's a great way to get a population estimate here. Okay, here's this is the trap, where the crews and then flipping. This is the recapture crew. It is a lot of fun. You can handle a lot of fish. And then this is a mess of a table, but actually, I don't know. So this is the overall population estimate that we get. We really only do this on one, but you do get this quite this range of different train sizes. So 2019 was a big year. 2020 was a really big year, but they run in April. So April 2020 is. We couldn't get a whole crew together to, it was kind of just those weird times, but yeah, I really wish we had a crew for that year. It's just but then in 2021 and 2022 did no run. And so that, you know, and I say no run, we had like maybe 50 show up that we like observed, which isn't enough to do in capacity, not even enough to harvest. And so there's a lot during those years, especially in 2022, we were just asking a lot of elders, is this normal? Do you remember seeing this? Or just don't come? And you know, I think that there's possibilities where sometimes they don't come to a river. But 2023, we were happy to see them come back. So yeah, so this is kind of just reviewing the marker capture. It's the challenges associated with it is have this active subsistence harvesting happening and involved in the forage fish. There's sea lions, tons of birds, everything's eating all the time. So it does meet all of the assumptions of a market capture study. So there's some flaws with that method. And we can really only do this on one river. And it costs a lot. We have crews, we have 2 crews out there, we hire about 10 people. They're there at the river from dawn till dusk every day. So yeah, it ends up being kind of expensive. We can only get data on the river. So this was a few years ago, well, I think 2017. We did catch per unit effort. It's kind of a reduced effort way to get some sort of population data prior to our eDNA, which we're going to get into. So this is on the Taiya Rivers. Yeah. Cool. Yeah. So catch per unit effort is another method for getting population data. These are all good. These are like kind of our early efforts in getting data going and getting some monitoring on the ground, seeing what's going on in the river. Yeah, so the egg and larval out-migrating. So there's larval out-migration is another population metric. So we're kind of, for a while, we're kind of like throwing anything at the wall and just see what would stick, basically. And just like, how can we get some data for this fish in all of these different rivers? We knew we weren't going to be able to do river capture and science. What else? What else we use? And so then this is eDNA. This must be. Am I stealing your slides?
That's okay.
Okay. Do you want?
Makes it flow.
Okay. Are you sure? But yeah, do you have anything to add? Do you have anything to this one? I'm not really sure.
Basically, I was just going to say we have this pretty.
Oh, okay. So this new method is taking water samples. So take these RoPack bags, collect a lot of them, so 3 per site that we're sampling, or 3 replicates, and and we do this eDNA or environmental DNA sampling. So this is kind of this new technique where we're actually doing this on a regional scale now. We should say that 2023 now. But so the just eDNA is taking that water sample and filter it down. And on that filter, we capture all the DNA of all the species that are in the water. So there's definitely it's kind of my fault. That's why I heard this. So you get all the DNA on there. So there's there's other species DNA in there as well. Then in the lab, we're able to just a single species pathway in there, and you can just look for how much eulachon or other species. In this case, we're looking for eulachon. No, it's non-invasive, expensive. and it just requires a technician to go out and collect water, filter it. So we can potentially sample many more rivers in a day. So yeah, here's the gist of it. We collect these water samples, filter them down, raining method of filtering, and then we won't filter paper in there. We extract all the DNA from those cells and run this process called digital droplet PCR. We all got a little more aware of PCR. But for this method of PCR, take our sample and put a primer in, and sample gets partitioned into like 20,000-ish droplets. And if there's eulachon DNA within one of those droplets, and so that's great, but we don't want to know just if eulachon are there or not. We want to gauge the size of the run in all of these different rivers. So that was our big question. Like, well, yeah, we'll be able to detect them, but can we detect change in abundance also through this eDNA method? So we'll go over how I think we are. This is just to show this is a positive control. This is the output from the ddPCR. So eulachon tissue sample, tons of positive droplets there. Good. This is when they're really rare in the system. So we're getting positive, some positive droplets, some that are not. A lot of them are not. And this is when they're really abundant in the system. We're getting tons of positive droplets. That's kind of what you want. You want that. You want everything to be positive, because there could be potentially more than one copy of eulachon DNA per droplet. So you want to get both positive and so I'm going to get heavy into the stats here. But this is just a comparison between the marker capture in the Chilkoot River and don't pay too much attention to this, but this is the eDNA kind of area under the just a way to gauge how much DNA is in the system. And so we see a really good relationship here for stat nerds, significant relationship. But yeah, so initially when we started this, yeah, it turns out move forward with using eDNA as a quantitative measure on eulachon populations and a variety of. Yeah.
Thank you for that explanation. I just think it's the coolest stuff. I think I said that it's at the last outreach. When I was in school learning about environmental DNA, they were only using it for detection. They were going into a pond and looking for a rare frog. And we could say, without having to capture one of these frogs, oh yes, this endangered frog is living in this pond. And now we're to the point where we're actually taking and getting robust information that's comparable to these complex statistical methods to getting a population estimate. And instead of taking 10 people and having them out there from dawn till dusk, just clipping and catching and releasing and clipping, me and Colton are going out there and we're grabbing water samples. And the worst thing that happens to us is when the Taiya River gets dirty and we got to filter in the lab for 12 hours. I mean, that's not bad really when you compare the two. Really just fantastic. So let's look at our actual sites here. We're looking at a little bit of an overview map here, right? So remember that I said wildlife observations are a really important part of our methodology. So we always have a couple of observation sites set up in our different places. So let's zoom in here to the Skagway River real quick. And for whatever reason, I decided to do this zoom. Here we are, almost there. Okay, there we go. So we have our observation site right here, right next to the sign that says, please stay 200 meters away. We're rogues, I guess. But it really is the best place for us to catch a glimpse all the way out. This is showing an extreme low tide on the Skagway River. And you can see the observation site is still going to be able to capture whatever is out here. So if we've got sea mammals that are out there that are munching on stuff, we'll be able to see them. And all the way up into the tidally influenced portion, we'll be able to see all the critters that are feeding them. And then we've got two sites. We've been doing two sites, I think, since 2020. We do a main river site, which is within that tidally influenced part of the river. So that's actually just downstream from where the tide really kind of butts up against that tidal influence. And then we do an estuary site. And I think the idea here is that we wanted to capture more of the run. If it's further down in that watershed, we're gonna capture more of that DNA. And then it also comes into play with the metabar coding, which is a very, you can explain that a little later.
I didn't put a slide up. We can talk about it.
Yeah, we can talk about it. Yeah, metabar coding. It's just a really cool thing. So we've got this observation site and we've got our 2 river sites. So we'll zoom back out so we can see them all. And one thing that you might notice here, after I put the labels up, is that the Taiya River has 2 observation sites. The reason for this is because the Taiya River is a much lower gradient, right? So the tidal influence is way up, almost all the way to the bridge there, right? And so when there's a low tide, this is again showing an extreme low tide on the Taiya River. You know, it's all the way out here. If we're at this observation site trying to see all the critters, we're not going to be able to see them. So we have this other observation site for when it's a higher low tide, so that we can capture at all different tide levels what all the animals are doing. And then you can see the estuary site much farther away from the river site compared to the Skagway River. Again, that has to do with the river gradient. We are doing triplicate samples, right, for quality control. We want to make sure that one sample, if it's way off from the other ones, we've got 2 more samples to compare it to, just so that we always know that our methodology is correct. We're also collecting an equipment blank so that we can tell whether or not we're doing any cross-contamination. We're dealing with DNA here, right? So everything's got to be super, super clean. Let's see here. What we got here? So here's some of our old, this is back in 2020, these are the Whirl Pack bags. And you can see, since we're doing triplicate samples, we've got this triple manifold for doing filtering. It's got a little vacuum pump attached to it to help draw that water down. When it gets really silty, this stuff goes super slow. I don't think I have an example of that, but here's some of the old vials that we would put our filters into. These are filled with ethanol alcohol, which ended up being incredibly difficult to ship, from what I understand, because it is a controlled hazardous substance. So this year we switched to something just a little bit different. This year we had these little silica balls and these little coin purses, and so we were preserving them with this, and so that takes away that need to ship this really caustic stuff. I love getting a package from seaplanes that says poison on it. It's just kind of fun, but we got rid of that this year. And then let's also look at me giving you the come hither look. This is pandemic times. We were filtering at home. But this is what it used to look like. This is a year's worth of, like, 1 season's worth of disposable plastic that we would have to employ to do this work, to make sure that we're doing it all very clean. I think every single person who worked on this project, it just killed us to look at this plastic. Like, we hid them away in boxes. This is the first time I actually put it all together in one place, and I cried. And it's just so sad how much plastic you need to use in order to do this stuff sanitary. We've got all the filter funnels, we've got all the Whirl Pack bags, packaging that comes in. I think last year for this outreach, we gave out filter funnels to people that came in so they could go home. They're really great for starting plants in. I had a picture up here. We were putting caddis flies into it with little jewels to make them build their little cases. Tried to find some uses for it. These make great bagel bags, by the way. Did about 5 bagels in the Whirl Pack bags. Works out really good. But our fearless leaders solved this problem in 2022. This is a yearly setup now. This is what we use. We use all glass filtered funnels. So we have to wash these and sanitize them in between each of the samples. We're using these reusable Nalgene bottles, so we don't have these Whirl Pack bags. The biggest waste that we have are just little pieces of paper on our filters. We take those out of their packaging so that they stay nice and sanitary. So it's a massive improvement. I mean, you go from that every single year. So we've got 4 years of this stuff built up somewhere. If anybody wants a filter funnel, we've got some for you. No problem. So it was just great to see it kind of change to this, right? So here we go into field season 2023. We took our first samples on April 7th in both rivers and started our wildlife observations simultaneously. I think we were supposed to do our 0 samples a little later in the season. I thought I remembered it being closer to the 10th or the 15th that we were talking about it, but we accelerated our timeline because there were observations from the community that there was this run of fish and people were calling them eulachon, right? So this is the very first one that was caught. This was in the Taiya River, and this was April 1st that this picture was taken. And then I went down and investigated, and I took this picture, I think, on the 10th. So sometime between the 1st and the 10th, there was this fish that was coming up. But we determined, based on the size, based on the morphology, and based on the timing, this is probably not a eulachon. Let's look at a eulachon compared to a capeman, right? Eulachon over here. Note the size of the eyeball. Note the position and the shape of the anal fin. And then compare that to this fish. Does that look more like a eulachon or capeman? Probably capeman. That's what we figured. It was also much smaller. And then I think also our results showed that we didn't see any eulachon DNA in the river prior to during this time period, but we did see an uptick in wildlife. So we were out there collecting samples just to make sure. So as the season went on, we got, all of the project partners were working all throughout this region. The project partners are communicating with one another. When fish are entering into a river, we find out and we tell each other what's going on. So the Berners Bay crew reported seeing confirming fish first on April 22nd. And then we got a confirmation of fish in the Chilkoot on the 25th. And there were people were basically thinking, based on the wildlife, that they had also entered the Chilkat probably a day or two before that. So we were anticipating that it was most likely we would see fish, either the 25th or the 26th. So we started gearing up and getting ready to go to daily sampling because it seemed like everybody else was getting fish. And unfortunately, the next thing that happened is things are going to show up. The municipality began pile drivings on the 26th and they continued on the 27th and 28th. There was a short little period where they weren't doing anything, and then on the 30th, when we started pile driving. This was loud. Anybody remember hearing this in town? It's incredibly loud. So if we're talking about a fish that has an aversion to noise, that could definitely be a problem. The timing is just, it couldn't have been worse timing for this to happen. So here we are. Here's the what is this? This is Skagway mughal counts. Mughals are kind of the main piscivorous bird that we're counting out there. It's the typical seagull that you see out there. And if you look down here, you can see, there's a couple little jumps and spikes, up into, the 3/4 of 100. But all the way up here, here's where that pile driving started, right? And we see everything is just nice and low. And then the day after pile driving stopped, we've got this big spike in wildlife. So just an interesting little bit of observation from that. I think also it's noteworthy to look at the isolation of those elevated numbers. Typically what we see is we'll see these big spikes and then we'll see the numbers kind of attenuate. But what we saw this time is we saw this big spike and then we just saw them disappear. So I think that that's interesting and noteworthy. Wildlife observations on the evening of April 30th, after the pile driving had finished, showed that we had this sharp increase in gull activity on the Taiya River. We didn't really get a whole lot of good pictures, but this was definitely one of the bigger populations of gulls that we had seen. And of course, this spreads out across the estuary. And if you look at the graph, you can see that reflected here. Note there on 30th of April, we have this big spike, but then notice also, see this kind of attenuation, this continuation of these high wildlife numbers, because the fish are still there, right? They're entering, we assume that they're entering into the river when we see these big giant spikes, but then they stick around because there's going to be carcasses coming down the river, right? They're going to be in the river spawning, so they'll be following to different spawning spots. So we don't see a sharp decrease in wildlife typically. So we never saw fish on the Taiya. We never actually got visual confirmation. And this is the first year that we didn't see any fish with our own eyes. Wildlife numbers continued to decrease. So we switched to every other day sampling and continued that through about May 24th when final samples were pulled and the season officially ended. So we definitely had a number of challenges this year when it comes to this non-charismatic fish that has no protection. We had people coming to us, is there anything that we can do legally to stop things like pile driving or motorized tours going right up the Taiya, right? There's really not a whole lot that we can do except try to appeal to the good senses of folks and say, hey, this is a culturally significant fish. I'm hoping that this project and more research will maybe afford us a little more legal protection for them. But for now, it's really unclear how these different factors affected the fish, except when we look at the data, we can move and be able to see a little bit more. So with that, would you like to show us some of our eulachon numbers? And I think for all of us today is one of the first times that we're seeing all of these data, which is exciting.
Oh, yeah. So, yeah, it's kind of, can't see the top there, but this is, I think this is 17, this is probably Taiya River, yeah, 17, 18, 19, 2020. So, yeah, so 2023 was definitely a lower, or, yeah, 2023 was definitely a lower year. Some of the previous years we've seen. 2019 is still kind of the standout banger year for a lot of the rivers in the Northern Canal. Yeah. It for that one, and then this is the Skagway River. And same thing, but we didn't have data in 2017 there, so this is 2018, 2019, 2020, and then the last 3 years. And so, yeah, again, 2019 was a big year for all of these rivers. 2019 this is a big year for all the rivers except for the Chilkat River. That was the year that the blasting was happening in the lower part of the Chilkat River. And so that is like typically a very, very big run, and probably one of the bigger runs in the Northern Lake Canal. It's just a big river, can support a lot of fish. And so when all of those fish kind of get diverted, all of our other rivers saw kind of gangbuster runs in 2019. Yeah, and then 2023 is low, but it wasn't, wasn't necessarily the lowest we've seen. So that's good. And then let's take a look at this is kind of tricky. Oh yeah, I guess I can, and here I can see what river it is. This is the Taiya River. Just can't see the label up there. This is the Taiya River. In the blue, we have this blue line is the eDNA concentration, and then the red bars are those that the daily mu counts. So you can see how the eDNA concentration also kind of tracks pretty well the gull counts. And so we did have a spike earlier than the eDNA spike. Probably a lot of gulls getting excited and showing up. Getting over there. And then, yeah, kind of a little bit different on the Skagway River. We had this quick peak, and the gulls came. Yeah, this is definitely much lower axes. If you look at the eDNA concentration, lower concentration on the Taiya River compared to on the Skagway River compared to the Taiya River. And then let's take a quick look at comparing as Ruben mentioned, we're sampling in the estuary and further upstream. So just comparing those for 3 of the years. We didn't do it in 2021, but 2020, 22, and last year we did both sites. So this is the Taiya River. We see the estuary and the river sample, like really tracking pretty nicely with each other. And this is, as you might remember, these are like further apart actually than they are on the Skagway River. So the estuary sampling location and the river sampling location are, I don't know in distances, but they're further apart than on the Skagway. Almost, yeah, there's a whole kilometer difference. So kind of interesting to see, as you'd expect, the estuary is slightly higher. If you're thinking about DNA, there's going to be more downstream. It's all flowing downstream. And there's definitely going to be spawning that's happening in between those locations that's being, detected at the estuary site and not necessarily at the river site. And then this is the Skagway River. And you see here, these ones are like not that far apart. We're seeing much higher estuary concentrations typically one of the years. 2023 is a little wonky. But I'm not sure what's going on there, but it's lower. But then, yeah, kind of to see more in the estuary, so that kind of, what that would tell you is that there's probably a lot of spawning that's happening in between those two sites, even though they're not necessarily that far apart. Yeah, so that is all the results we have.
And then, yeah, if anyone wants to talk about metabarcoding, I'm happy to, but.
Yeah, what is metabarcoding? What are we doing that for?
Yeah, so metabarcoding is where we do that on mainly just on these estuary sampling sites. And as I had mentioned that ddPCR, we're looking just for eulachon DNA, but with metabarcoding, we can kind of get this more like a shotgun approach where we can see just like what species are there and hopefully in what relative abundance. So metabarcoding we can use to kind of track some of those wildlife trends through eDNA, see how wildlife are congregating around the eulachon population responding events. Yeah. Any questions on any of that? Make Ruben answer.
I'm gonna say something. Sitting here listening
Yeah.
and how the hooligan, it's the hooligan, the chum salmon, silver salmon, and also, the millfish. But it has died down in Skagway. I've been here since 1954. 54, the Skagway River was just full of hooligan. You can go all the way up to the bridge and just take a 5-gallon pail and you'd have it full by just one dip. Getting it. And so probably when we had the high water in 68 or 67, 68, so that changed the whole river because it almost overflowed the airport. It was only about a foot before the water went over, the river went over the airport. And not only that, it's when White Pass started hauling ore, the whole waterfront had changed. So that changed the whole system, that waterfront. You know, I don't know if it's hooligan or like king salmon, they come back, every 5 years. And then they know exactly where to go to lay their eggs and stuff. I'm not a biologist, I'm just guessing. And so that all changed. So, the hooligan, has slowed down and so in the 60s, there was hardly anything that ran up Skagway River. Most of it went up Taiya River because there's no industrial stuff done over there. And also with the high water, the White Pass changed the river up at 5A Bridge because it was washing out the embankment from 5A all the way down to the bridge down at Skagway. So that changed. And also, when they built the Klondike Highway at Lyersville, there was a creek there that used to be, the dollies used to come up, chum salmon, silver salmon. And as soon as they changed that, then they couldn't run it up there. The Pulling Creek, it had dog salmon every year coming up. I never seen any hooligan, but mostly dog chum salmon running up, and they go all the way up, almost up to the shops. And so a lot of it just is, and then White Pass changed the culverts and they blocked the water and stuff, so that changed it too. But every time they touched the river, either the state or White Pass changed it, the runs just died out. So it's
Yeah, habitat's
When you
definitely.
look at your chart, you said 1950s, 54, the hooligan was really strong in Skagway River. Or 55, but I came in 54. And it was strong. I mean, the river was so black, you couldn't even see the all the way across the river.
Yeah.
And the river mostly ran over towards the airport. And then of course they changed the airport to fishing games. Oh, there's no fish that run up Skagway River. Yeah, like, oh. It's nice, the way the airport is now, it's a lot safer. But for running the fish and stuff that run up, king salmon in the 50s, you've been out fishing, you're lucky to catch a king in 8 hours. Or even longer than that, I go out every day, go out fishing, I'd be out there for 8 hours and maybe catch one or none. Then they started the fish hatchery, that brought the kings back, but
Yeah.
And then they did the same thing, they had the hatchery here. they gave some of the eggs to go up to Chilkoot or Chilkat River to build that stock up.
Mhm.
And I don't know how it worked down there, but, in Skagway, the state ran out of money. They complain about the king salmon. There's no king salmon. And so we'll shut down Haynes, we'll shut down Skagway, so the orcas can have salmon. And then it's the orcas that they release from the, whatever it's called, down in California. And they're dying because there's not enough feed for them, because they don't know how to do it, because they're hand fed. So it's just a round and round circle.
Yeah,
Yeah, so, you can't blame it all on that fishing. You can't sell by them fishing too close to the rivers where the salmon run up or whatever run up.
Mhm.
They push their nets too close. In May, herring used to come here. Every May, you'd have a whole bunch of herring in the boat harbor.
yeah.
And I haven't seen any for quite a few years.
Oh, wow.
Maybe not, but a few years. And they used to lay their eggs all on the boats.
Yeah.
I would have to take their boats and put them up on the ramp and pull them down because they're outboard motors, and they get, what their
Yeah, you know,
water pumps out and stuff.
yeah, it's definitely interesting. I think that that's a good point, though. Like, there's
Yeah.
this like shifting baselines
Right?
kind of syndrome we talk about a lot with fisheries management, or even just kind of a lot of
So there's,
wildlife
White
management.
Pass built a new dock and they're all that pile driving,
Mhm.
steel pipe.
Yeah,
And then they extend the dock down here, pile driving again.
yeah, yeah,
And now they're doing the work now, but there's nothing coming in, but
yeah.
they have people watching,
Yeah.
but, what can you do by having somebody out in the shore watch? Oh, yeah, there's something out there, better spot, I'm seeing, no. But anyway, I just listened and I thought, way back then when I first came here,
Yeah,
how it was so nice to just go get hooligan or go get herring or go get dog salmon and chum salmon or something. And,
Yeah.
always used to, always night and kids were growing up, they'd go over by the big rock and cast out there and catch a dollar and maybe it's their very first fish they've ever caught,
yeah.
And now, because they used to follow the king salmon. And just to get their eggs, and that's what they do. In Point Creek, they follow, the dollies would follow the salmon up and they lay their eggs. They start eating them. Yeah, anyway, that's my 2 cents.
Yeah, thank you.
Thanks, Andy.
Yeah, any other questions?
Also, mention a positive note after Andy,
Mhm.
because I just came across this like a month or two ago. I think it was like August when this came up. The Port of Seattle released that they found 73,000 juvenile salmon in their estuary habitat after they started doing some habitat recovery work in that area.
Oh,
I think it was like the first time in years in that port. So it's not a completely lost. There's
Yeah.
probably some habitat restoration that could potentially happen one day, even though Ruben had that port picture of the Duwamish port. I think it's in that area where they did some restoration and had 7,300 returned.
yeah.
They're finding this in Washington, all the different places where they're reconnecting this old habitat that's either behind a dam or back where I'm from in the Skagit Valley. They've turned in the delta of the Skagit River into this big farmland. There's an island right in the middle of these two branches called Fur Island. And they did the numbers, they did the math, right? And they figured, well, actually salmon is going to be more lucrative in the long run through ecosystem services than having this farmland. And so they reconnected this old estuary that was behind dikes, and instantly within a few years, it was being utilized by all these species of salmon. Same thing with the Elwha Dam. They take the Elwha Dam down. It's been 100 years since fish have gone up past Glines Canyon, and now it's occupied by, I think, all 5 species of salmon. It's just amazing how quickly and how resilient these these critters are at going back to these places where they've evolved to live in. And as soon as we make conditions better, we're finding that they reoccupy them very, very quickly.
Then they'll lift their net. Fishermen go to get them pork mount again.
Right.
Big gill nets.
I just noticed we've got some questions in the chat.
Oh, there's questions in the chat. Oh, good.
So Paul in the chat has asked, why adaptively speaking, do Saak have such a high oil content? And we didn't mention herring, but they're significant in this region as well. I would say probably because they're a deep water fish and a cold water fish. They need to have that insulation. And it's also based on what they're eating. So if they're living, you know, in the northern Pacific, you're gonna have higher oleic acid in your phytoplankton. And yeah, we don't mention herring. We don't really deal with herring here. The herring used to show up in Skagway, but in Skagway, I think the eulachon was culturally more significant. Perhaps the herring played a part as well, but I think we hear more stories about the Saak versus the herring.
Yeah, I don't have anything to add to that really. Yeah, herring are definitely still very important for, and they kind of, in Haines area, they show up usually right before the hooligan or the Saak do. But yeah, herring are also very rich in oil. And I don't know their oil content off the top of my head, but
I'd imagine it's comparable to at least Cape Woman.
yeah, I don't think so.
And Paul also asked, oh, go ahead.
Yeah, I tried to look in for help, I don't know, I never caught one.
No.
Maybe it has to be alive.
Oh, sure.
Yeah.
And then I also put, hooligan oil on herring, trying it, because somebody gave me a jar of it. Well, I never found one.
No.
It must, I guess, you have to talk to it.
Yeah.
Paul's also asking, I think it was when we were talking about the different factors that are causing the decline. We talked about dams. Were they really going that far up the rivers past where the dams are? And I would say on the Columbia River, yes, because the Columbia River is such a low gradient. They were putting dams up way down in the main stem that probably, I would imagine that they would have been able to travel it past that.
Yeah, I mean, we see them, they do go, you know, in the Chilkat, even like on a big year, we'll see them go at least 8 miles upriver. So they definitely go beyond tidal influence. And so, yeah, I think the Columbia, there's definitely potential for them to interact with dams.
And Jennifer asks, how do cruise ships affect them since they're coming earlier in the season? We don't know. We should get some funds and put some hydrophones out there and get some cameras and start taking some footage and find this information out, because that would be important information to know. Is the cruise ship affecting the number of fish that are coming up either down in Haines or here in Skagway? That it very well could be, but we just have no data on that.
Yeah, and there's some studies that are being done now in British Columbia looking at how sound avoidant eulachon are in related species of eulachon. And so that's like kind of been this missing piece that we don't have this like concrete data. Like we have these observations, we have traditional knowledge, you know, regarding how sensitive they are to sound, but we haven't had like a piece of data that we can go to agencies or, you know, and speak to how, you know, documented how sensitive they are. So I think that will also kind of help. And then if we can get some sort of, you know, levels of sound, then, you know, being able to detect the sound coming off of cruise ships and how that might be influencing them. So I think that's coming. I'm hopeful that we'll be able to say something more significant about how it's impacting them. It is something we're observing over and over again, but
I have faith as well as traditional ecological knowledge gains more of a footing in the scientific community. I would argue that 10,000 years of observation definitely trumps a couple of studies that were narrowly focused on 1 or 2 things. You know, if you have this consistent story coming across cultures that's saying the same thing, we found that traditional ecological knowledge is very, very, very important and completely worth taking seriously. So hopefully that will be a shift that we see. Paul asks, if they're not directly commercially important, are they a keystone species as a forage fish?
Oh, yeah, definitely. So there's been studies on sea lions. The sea lions, stellar sea lions were listed as endangered in Southeast Alaska recently. I forget what year they were delisted, but it is in the last 10 years they got delisted, I believe. And they found that, and this is a study that happened right in Southeast Alaska, that this impulsive eulachon, this like high oil content, huge fat resources, is super important for the stellar sea lions before they give birth. Because then they don't eat for a couple months, and it's a lot. Having this reserve of fat allows them to actually nurse their young while they're not eating for a couple months. And without eulachon, they won't be able to actually feed their young to survive. And so it provides this really key food resource at a really critical point in stellar sea lions life cycle. And that's just one example
Yeah. I'm
among
sure
many
it's-
of like of how important they are
Yeah.
as an ecological species.
Yeah, I'm sure, well, like we saw with other forage fish, you take that piece out from the trophic levels and you start to see these cascades. So seabirds and, sea mammals, you'd probably see a pretty significant impact all up and down trophic levels if you removed that. So yeah, keystone species, I would say.
Yeah.
Here. Also mobile, Chilkoo's not far away. Motorboat tours, the hovercraft tour is a new tour run by locals here in town. Scouts come through over 2 weeks before the rest of the run. Oh, you're probably looking at that spike in the eDNA data. Yeah,
Yeah, so I would potentially say like some of these are some scouts, these early peaks that we're seeing. Probably some scout DNA. There's also traditional knowledge and we've seen it documented, but there is a January, February run that does happen. It's usually much smaller. And so there's even, some lingering DNA that's in the river even when we're taking our zero samples. So.
And also the population of seals has grown
Mhm.
since they can't get $2 for another.
Yeah,
Because every once in a while in the winter, somebody will say, yeah, when the ice would freeze over in Taiya, that they'd see 200 seals. They're just waiting for whatever.
yeah.
And sea lions, seals, humpback. I don't know if humpback will eat them, but sure will.
Oh, yeah, humpbacks definitely are eating.
Oh, they will?
eulachon.
Oh,
Yeah,
oh,
yeah, in
that's
big
why they
numbers.
hang around.
Yeah.
I always thought they were just after plankton.
Yeah, we see them eat eulachon.
Interesting.
Yeah, they'll even like in Lutak Inlet, we've seen them just kind of like run almost like along the shoreline. If it's like
Going past them.
just like get really close to the shore.
that's an efficient way to fish.
Yeah.
Having a mouth that's 20 feet wide.
Yeah.
Oh, yeah, and then Jenny News says maybe they can do the pile driving earlier, like in March, before hooligan are in the area, and that is definitely a request that we've put out there, is that any other time of the year, it's like we understand that it's part of the infrastructure here, that infrastructure needs to be updated, but it's just a small period of time that we're asking that we give some consideration to this non-charismatic fish, right? So hopefully it sounds like they are, that isn't the plan for the upcoming. They're gonna stop by the beginning of April. So hopefully that will follow suit, and we will have another great run of eulachon this next year.
Yeah. Any other questions or thoughts?
You guys have been so great hanging out for an hour and a half. Holy moly. It's been a little bit of time. Do appreciate you coming out on such a rainy evening. Yeah, thanks again. We'll see you the next time.
Thank you.
Yeah.
All right, I am going to end the recording.
Oh,
Mm.